All right, welcome to the next set of lecture as part of chapter 10, which is all about membrane transport. And in this lecture, we're going to talk in a lot more detail about passive-mediated transport. So in this chapter, just to summarize, in the first part, we talked a little bit about the concepts behind the thermodynamics of transport. In this lecture, we're going to get more into passive-mediated transport, talking about a number of different transporters, some of which are proteins, some of which are porins, but not all. And once we're done with that, in the next video, we'll then talk more about active transport. But first, let's get back into passive-mediated transport. So just as a reminder, as we learned in that first lecture, passive-mediated transport is helping out with movement that is already energetically favorable or spontaneous, meaning that a molecule or an ion is moving down that electrochemical potential gradient. The speed of this movement is just facilitated by a protein or whatever the transporter is. So a lot of the transporters are proteins, but again, not all of them. And this movement would happen on its own, but it would be slow, which is why we have our transporters helping us out. So the first type of passive-mediated transport we'll consider are ionophores. And ionophores, like the name suggests, help out with the transport of ions across membranes. So ionophores, they move ions. They are small organic molecules. They resemble proteins, but they are not proteins, and we'll see why in just a little bit. And they are generally of bacterial origin. And their general function is to equilibrate ion concentration. So essentially, bacteria can use them as sort of antibiotics. If you can insert an ionophore into a competing cell's membrane, you can help to equilibrate the ion concentration across the membrane of that competitor cell. And that can ultimately stop a lot of different processes that are going on inside the cell and not allow the cell to respond to various signals from the environment. And ultimately that loss of that ion gradient can kill a competing cell. So ionophores you can think of as having that antibiotic effect because they're equilibrating ion concentration. So how do they do that? So we consider ionophores as carriers. So they, in this diagram, this big kind of pink and red molecule is the ionophore. This molecule then binds to a small ion. Like we mentioned, this is going down an electrochemical gradient. So generally it'll bind an ion on the side where there is more of that ion, and then it'll bring it over to the side where there's less of it, right? So that's that passive facilitated movement. And so then essentially the ionophore is going to transport the ion through the membrane. Like we see here, it's going to flip from one side to the other, and then once it reaches the other side, that ion will be released. And at that point, generally the ionophore will return to the original side and keep moving ions across until ultimately all of the ions are equilibrated on both sides of the membrane. So one specific example of an ionophore that we're going to consider is called valinomycin, and this ionophore transports potassium. So we can look at the structure of valinomycin here. It is a cyclic molecule that consists of a number of amino acid-like molecules. But it's not a standard peptide in the way we learned about them before. So first of all, as you can see, there are both D and L amino acids. So in this case, we have both L and D valine. And then we also have some non-standard amino acids that are not quite amino acids. And these are the dehydroxyisobularic acid as well as lactic acid. And so if you recall from when we talked about amino acids forming peptides, we talked about the peptide bonds that form. And these are essentially amide bonds between a carbonyl group and an amine group of a neighboring molecule, monomer, as part of this polymer. But you can see in this structure that we actually have a number of different linkages that are not peptide bonds. We have some ester bonds. So we have one here between L-valine and dehydroxyisobularic acid, as well as one between D-valine and lactic acid. So this goes to show you that it looks a little bit like a peptide, but it's not quite a peptide, so we can't call this particular carrier a protein, right? Like we mentioned, it is cyclic. And the way that it forms its three-dimensional shape is, we can see over here at this diagram at the top. And you can see that in the center here, in blue, we have our potassium ion, and it's positively charged, right? And so it's going to happily interact with all of these with the backbone part of this valinomycin molecule. So all of these oxygens that are part of the backbone and some of the nitrogens that are there as well, those are going to help that relatively negative charge is going to help to stabilize that positively charged potassium ion. And then we also have, as part of our valinomycin molecule, we have all of these sort of side chains, which here we can see they're all, for the most part, very hydrophobic, right? They'll resemble that valine side chain, which is all hydrocarbon, it's all hydrophobic. And you can see those hydrophobic sort of V-shape side chains sticking out all along the outside of this molecule. And so you can imagine that's going to help us out when we consider how this looks in the cell membrane, right? So we want the outside portion of this ionophore, valinomycin, to have that nonpolar sort of hydrophobic region that's going to interact with the fatty acid tails that are part of the cell membrane. And then we only want that central part that's interacting with potassium, the ion, to have that relatively negative electron dense region. So let's now think about some different types of ionophores. So we just considered one where it's simply carrying the ion right through the membrane. But we also have another class of ionophores called channel-forming ionophores. And these ionophores form, like the name suggests, a channel through the membrane. So they span across the whole width of the membrane. And we can compare that to our first one, valinomycin, where it doesn't span the entire membrane, it has to carry itself across. Our channel goes all the way through the membrane from the outside to the inside of the cell. And these types of channel-forming ionophores are also often transient. They're not always open, and so they're not always allowing ions to pass through them. But when they're open, they do allow ions to pass through. And again, we're talking about passive-mediated transport, so these ions are going to be going down that electrochemical gradient. So an example that we can think about here for our channel-forming ionophore is gramicidin. Gramicidin is a channel-forming ionophore that helps to transport monovalent cations. Gramicidin itself is a peptide. It's a real protein. It's linear, and it's made of 15 amino acids. And these are in alternating D and L amino acids, so they're not the sort of standard amino acids we might think of in our own bodies. But that's what gramicidin is made of. And we can see from the shape of it, in particular in the spinning diagram, that it forms this beta helix. So what is a beta helix? It's a little bit larger, it's looser that you can think of than an alpha helix, and that makes it such that the space in the middle forms a large enough pore for something to pass through, which we can see in this top-down diagram shown at the top of this slide. And so this beta helix, just like we saw with alpha helix, we have our side chains projecting away towards the outside of the helix. And the inside of the helix is lined, again, with the backbones of all those peptides. And so if we recall, our peptide backbone, again, is fairly electron dense. And so it's going to help to interact with those cations, right, and help them move down the middle. Whereas the outside side chains, you can see, are going to be interacting with those fatty acid tails that are part of the cell, the inner part of the lipid bilayer membrane. And so that's where you want those hydrophobic side chains to be interacting. And then something to note here is that you actually need 2 gramicidins to actually span across the entire membrane, and they'll stack on top of each other to make that happen. So moving on from ionophores, now we have a class of transporters called porins. And porins always form beta barrels, which may be familiar to you from a few chapters ago when we were talking about protein structures and motifs, and beta barrels are one of those motifs that we see a lot. So beta barrels, or porins, tend to be, let me start that over. Porins are made from beta barrels. Porins themselves are large and multimeric, so you'll often see multiple beta barrels multimerized with each other, which we can see in the bottom part of this diagram. We have the top-down view, where we have 3 different porins that are all adjacent to one another. They're all colored in differently, and you can see each one has its own pore where something can pass through the center. So where do we see porins? They are frequently found in the bacterial outer membrane, and we also see them in organelles, in eukaryotic organelles that have bacterial origins. So this includes mitochondria and chloroplasts. So porins are able to transport larger molecules or ions than ionophores, partially because they tend to have larger pores in the middle. And porins are essentially able to discriminate what they're transporting across the cell membrane, both by the size of the opening as well as the side chains that are lining the barrel. And so this can help to facilitate the movement of certain sized and charged species, but not others. And that will help porins be discriminatory about what they choose to let pass back and forth across the cell membrane. So now let's talk about a selective porin, and the first example we're going to use is maltoporin. So maltoporin is found in, like a lot of porins, in the outer membrane of bacteria, and it's selective for transporting only maltodextrins. And maltodextrins are just oligosaccharides that are derived from starch. So as we recall, starch is a chain of glucose molecules, and when you start to break it down, you can end up with these oligosaccharides that have anywhere between 2 and 20 glucose molecules. And so we have this picture of just the repeating glucose monomer. We see that it has this alpha facing hydroxyl group at the anomeric carbon, and it forms these alpha 1,4 glycosidic bonds with its neighboring glucose monomers to make these oligosaccharides. And something interesting about these maltodextrins, because of this bonding, especially these alpha 1,4 bonds, glycosidic bonds that link the monomers together, they actually form this very distinctive curved shape. They are not linear the way we might sort of assume based on the way we draw these oligosaccharides a lot of times in a textbook. In real life, they're actually this interesting little curve shape. And this is something that maltoporin takes advantage of to help it be selective for only maltodextrin. And so we can see that in this diagram of maltoporin. So maltoporin is able to select specifically for maltodextrin because it makes this funny little helical shape, this sort of curved shape. So we can see in this picture, in the green and red, we have our maltodextrin glucose oligosaccharide, right? And it has that funny little curved shape. And then there's a corkscrew-shaped path in the maltoporin itself that's made from a number of different residues, amino acid residues that are on the inside of that porin. And we see that here in the yellow and blue is that sort of corkscrew-shaped path. And the amino acids in that path are a lot of, we got some tryptophan, we got some tyrosine, we got some phenylalanine. And so as you recall, a lot of those are actually very bulky and hydrophobic type of residues, which you would think would not interact well with a bunch of glucose molecules, right? Because we talked a lot about how glucose is very soluble, it's very hydrophilic. But something to keep in mind is that the glucose backbone, as you know, and as you can see here, is hydrocarbon, and it has all these hydroxyl groups attached to it. And so the hydrophobic amino acids that are lining this porin are able to sort of interact with the hydrocarbon parts of the glucose molecules. And that sort of helps to form, that sort of is going to help facilitate the glucose sort of being able to slide through. The glucose, or the maltodextrin, is not going to stick a lot to these hydrophobic residues because they'll want to repel each other a little bit, and so it'll help the maltodextrin kind of slide right down that little corkscrew-shaped path. Another thing that makes maltoporin very selective is the central pore diameter. It's very small. It's only 5 angstroms, and so it's a very selective and specific fit for maltodextrin molecules. And so, like we mentioned, this hydrophobic slide of aromatic side chains that we saw here, all these tryptophans, tyrosines, phenylalanines, they are going to interact with the glucose and form that greasy slide. So you can imagine the glucose sort of slipping down that little corkscrew-shaped slide, almost like a little tiny amusement park inside of the maltoporin. And so, like I mentioned before, the polar and charged side chains that are so, like, for example, tyrosine is a little bit polar, parts of its side chain, and so those polar parts of the maltoporin are able to interact with the hydroxyl groups on the glucose to help stabilize it, but the hydrophobic parts will help the glucose kind of slide right through. So that's maltoporin, a very specific porin. That's a beta barrel. Now we have another molecule that has porin in the name, but very important to note that this is not a true porin. And why is that? So if we look at this picture, recall that porins are structurally made from beta barrels, and this particular structure that we can see here is not beta barrel. It actually has a lot of alpha helix character, which you can see. So let's talk a little bit more about this molecule, which is called aquaporin, but remember, not a porin. And what is its job? It's to transport water. So we did talk about on the very early slides how water can, for the most part, freely diffuse through membranes, cell membranes, but it's not necessarily fast, right? That's the whole name of the game here with our passive-mediated transport. It's making spontaneous things go faster. So aquaporins do a very, very good job of allowing this very, very rapid transport of water across cell membranes. And it does this very quickly, and it's also very specific to water. So where do we see aquaporins in the body? We see them in the membranes of cells that are all involved in the rapid transport of water. Makes sense, right? So in tissue and organs like your kidneys, your salivary glands, and your lacrimal glands. All of these are involved in moving water across, and so they're going to need a lot of aquaporins to help facilitate that very quick transport of water. And like I said, aquaporins are very specific to water, so they actually exclude all ions, including protons, which is remarkable in a sense, right? Because protons are very, very tiny. And so if you imagine some of these porins, they're able to exclude based on size. But aquaporin, how does it exclude protons? If you had to fit water, right? Water is bigger than a proton. How do we exclude protons? So we'll talk about that in just a moment when we talk about the functional aspects of the aquaporin. But first, let's talk more generally about the aquaporin structure. So aquaporin is a homotetramer, and so we can see that in this diagram on the side. So each of the different subunits is drawn a little bit differently, but you can see that there are 4 of them. We have this sort of space-filling diagram shown in purple. We have this, the different color alpha helices drawn over here. Let me just start numbering these. We have our one that shows all of our different types of atoms, and we have another one showing our alpha helices. So all in all, we have these 4 total subunits, and each one has its own pore. And so in what I labeled subunits 1 and 3, the pores look very large, but if you actually take a look at those other ways of representing the structure that are labeled 2 and 4, you can actually see that the pores are very, very small. And so that's something to keep in mind as we go through this, which makes sense, right? Water is a very small molecule, and so one of the things aquaporin can do is exclude things that are larger by having a small pore. And so within each of these subunits, there are a number of different alpha helices. So each subunit contains 6 transmembrane alpha helices and 2 shorter alpha helices. And they each have a specific job when it comes to helping to transport water. So let's look more about that. So how does aquaporin actually transport water and water only across the membrane? So much of the pore of aquaporin is actually hydrophobic, and this allows for rapid passage. So we can think of it almost like we thought about that greasy slide in our maltoporin. For our aquaporin, we have this a little bit of a hydrophobic nature, so water won't spend a lot of time hanging out and interacting with those hydrophobic regions of the pore. The pore itself is hourglass shape. At its narrowest part, it is only 2.8 angstroms wide, which is very, very tiny. And then we have, and part of what allows so that's at this size restriction area, that's the smallest part, 2.8 angstroms. We also have this, the electrostatic repulsion, right? Like I mentioned before, for the hydrophobic regions of the pore that allow for the rapid passage of water. Sorry about not pointing that out before. So the next part we had to think about is how can we be specific for water? And so part of this is through these very specific amino acid residues that are in the relatively narrow part of the pore. And there are arginine, histidine, asparagine included here. And so hydrogen, and all of these have polar or charged parts that are able to hydrogen bond with just one water molecule and completely surround it. And this helps to separate one water molecule from all the other molecules that are present. And so, and this helps to specifically sort of pick and choose because of the exact spacing and size, as well as the charge interactions, this allows it to be very selective for only water by arranging it in this very particular way. You can see this particular water molecule that's been separated from all the other water molecules in the area and is interacting with all of these different side chains and backbones from the amino acids that are lining the pore here. And so this helps the aquaporin to be very specific for water molecules, and it also helps it to stop from forming a proton wire, or in other words, to prevent proton jumping. Because like we mentioned before, protons are really tiny and they are charged, right? Just like water has that polarity to it, protons are charged. So you might think that protons can kind of sneak their way through this porin. And one way that we prevent that from happening is having the water molecule reoriented about the dipole and separated from all the other water molecules. So let's talk more about this proton jumping that we're trying to prevent. And so we'll see how proton jumping works, and then you can see how separating the water molecule from the other water molecules helps to prevent this from happening. So let's consider we have this aqueous solution here. We have 4 different water molecules. Each of these water molecules is hydrogen bonded to its neighboring water molecule. And so we have in this drawing our covalent bonds are drawn in solid black. Our hydrogen bonds are drawn in those dashed lines. And so that's great, right? That's sort of normal. Water molecules love to interact with other water molecules. We have all these charge-charge interactions. We have these linear bonds that form. Great. So now imagine we have a proton that is in this solution. So a proton being charged is going to want to start to form a hydrogen bond with one of the oxygens that's part of a water molecule. And because of the sort of relatively sort of free movement of electrons and sort of the different attractions that we have here, that hydrogen bond between the proton and the oxygen can become a solid covalent bond, which we see here. And once that happens, now this particular water molecule with the central oxygen has now become a hydronium ion. Right? It's an oxygen that has 3 covalently bonded hydrogens, so we have a hydronium ion. And so because of the sort of relatively transient nature of the covalent versus hydrogen bonds, we can sort of imagine the changing around of some hydrogen bonds to covalent bonds. So let's picture how that would work. So now we have our regular old hydronium ion. We don't have a free proton in our solution anymore. The next thing that might happen here is, so I just want to make sure you're clear on what's happening. So this is our hydronium ion. It's H3O+. But this is relatively unstable. It's going to want to form back into water. So now what might happen is one of these hydrogens will now, instead of being fully covalently bonded, will now have more of a hydrogen bond character, which we see here. So we have this covalent bond has now become a hydrogen bond. And so then we can imagine that kind of goes down the line here. So now we have this proton that's kind of trying to form hydrogen bonds with both of its neighboring oxygens. And so maybe as a transient nature, now we have a covalent bond forming between that newly freed proton and the next water molecule. And so now we have, just like before, we had our first water molecule was a hydronium ion. Now our second water molecule is a hydronium ion. And we can imagine a similar thing happening where this hydrogen that's now kind of bonded to both of the neighboring water molecules, maybe it'll lose some of the covalent bond character to water number 2 and gain more of a covalent bond character at water number 3. And now our water number 3 is a hydronium ion, and so on and so forth. And so now you can imagine one of those protons might now start to form might start to loosen its bond to water number 3 and start to form a stronger covalent bond to water number 4, making water number 4 now into our hydronium ion. And then ultimately at the end of this, you will have one of these hydrogen protons start to loosen up its covalent bond and form a hydrogen bond, and ultimately maybe free itself as a sort of proton in solution. And so all this happened, all this changing of the nature of all these bonds, but none of these atoms actually moved, right? We had here, excuse me, I'll just make this a different color. So here our original proton joined It did what we call proton jumping by forming these alternating covalent and hydrogen bonds throughout this series of water molecules and this network of water molecules. And ultimately, now we ended up with a free proton all the way on the other side, even though, again, there was no movement. And so you can imagine, if we're thinking back to our foreign, right, if we have all of our water molecules able to sort of touch each other and form these bonding networks with one another, that would allow for proton jumping. But because our aquaporin separates the water molecules from their neighbors, it prevents this from happening. So that's enough about aquaporin. Let's now move on to other types of transporting proteins. And so now we're going to consider transport proteins that help to move molecules through a conformational change. So before we were looking at porins, and these are able to exclude or include molecules based on some properties of the molecule, but they, for the most part, didn't undergo a lot of conformational change. The next set of proteins we'll consider, these transport proteins, are going to use the conformational change to help move molecules across that cell membrane. So the example and the types of molecules that can be moved in this way tend to be amino acids and other small molecules. And so one of the examples that we're going to consider for a transport protein is the GLUT1 transporter. And this transporter, like the name sort of suggests, transports glucose. And it does this across the plasma membrane of red blood cells. And its main job is to just equilibrate glucose concentration, and it can go in either direction. And so, once again, as a reminder, this is passive-mediated transport. Molecules are always going to be flowing down their gradient, down their electrochemical gradient. And so, again, this is going to mean that this transporter is going to equilibrate the glucose concentration, because the glucose is going to flow in the direction towards the lower concentration of glucose. So how does this work? So we can imagine the cycle sort of starting over here. The glucose transporter is open towards the outside of the cell, which is shown at the top, and a glucose molecule is able to fit into this opening. Once glucose binds to the protein, then we have this conformational change that happens here as we move along this way, where the glucose molecule sort of fits into the middle portion of the transporter, and the transporter closes. The glucose starts to move through the membrane, right, from the outside towards the inside in this example. And then finally, after the full conformational change, now the transporter is open to the inside of the cell, at which point the glucose can exit the transporter. And then finally, we have this recovery period where the transporter will go back to its original conformation where it's open to the outside of the cell, at which point it can pick up more glucose and bring it to the cell's inside. And again, this can go in both directions. In this example, we're going from outside to inside, which is usually the case. Usually the cells are taking up glucose from the outside to then use the glucose to create energy, but not always. So let's think about transport proteins in general. So we just considered a specific transport protein called GLUT1, and this is essentially a uniport protein. It takes one molecule and it moves it to the other side of a cell membrane. So that's what we have with uniport. It goes in one direction, either out or in. Another type of transport protein, the other two types, I would say, can actually move more than one molecule at a time. And these are called symport and antiport. So symport transporters help to move two molecules in the same direction together at the same time. So here we showed this red molecule and this green molecule both moving down this way at the same time. And that's in contrast to antiport, where you have one molecule moving in one direction and a different molecule moving in the opposite direction at the same time. And once again, this is essentially all passive mediated. So for symport, both of these molecules are moving down their gradient. For antiport, both the molecules are also moving down their gradient, but one molecule might be present in greater concentration at the opposite side of the cell as the other molecule, and that's why their gradients are going in opposite directions when we're thinking about passive-mediated transport. So let's now actually switch gears a little bit and start talking about ion channels. So we talked a little bit about ionophores, which help to move ions. But here we have ion channels, and these selectively transport ions, and they are transmembrane proteins that go all the way across the cell membrane. And these tend to be pore-forming proteins, where the ion can travel down the center of the pore, either into or out of the cell. And ion channels are very important. They are required for maintaining the osmotic pressure balance inside and outside of the cell. They're important for signal transduction, which we'll talk a little bit about a specific example of that later in these slides, as well as for just maintaining the membrane potential. So we need to maintain a certain electrochemical gradient for a number of different ions and other small molecules in our cells to help keep everything functioning properly. And so we rely a lot on these ion channels to help maintain that homeostasis. And so that's their function. So now let's think about how their function can be affected by their structure. So the structures of these ion channels helps to make them both very selective and very fast at moving the particular ions that they're transporting. And in addition to that, these channels have the potential of being gated, meaning that they are sometimes open and sometimes closed, and they can be gated by different characteristics. And so we're going to look at two different examples of ion channels. First, we'll look at the KCSA potassium channel, and then we'll also look at another potassium channel called the KB potassium channel, but this one is a voltage-gated channel, and so this channel is regulated by the voltage, the membrane potential. So let's take a look at how these channels work. So first, we'll consider the KCSA potassium channel. This potassium channel is originally found in strep lipids. So it is a homotetramer that has one total pore. Each one of the monomers of KCSA potassium channel contains two long and one short helix. And you can sort of see some of that in this diagram where we have some of these helices in the inside and the outside. And it also has this selectivity filter that helps it specifically only allow potassium. And how do we know which direction the potassium is going to go in? And that, again, we're passive-mediated transport. We're only thinking about going down the gradient. So if we consider in a normal cell, there's a lot of potassium inside the cell and very little potassium outside the cell. And so these KCSA transporters are going to let potassium leave the cell, which allows them to travel down the gradient away from the high concentration inside the cell towards the lower concentration outside the cell. So how does that work? So first, let's consider the structure of this KCSA channel and how that leads to its function. So first, we have the cytoplasmic opening. So that's down here drawn at the bottom. And so we have the opening down here. That's the entrance for potassium to leave the cell, right? It's going from the cytoplasm to the outside. So ultimately, it's going to be traveling up this diagram. If you can imagine, the cell membrane is kind of over here. And so on the cytoplasmic side, potassium is able to enter. It's a fairly small diameter entrance. It's only 6 angstroms. And it has a number, in that location, there's a number of anionic side chains, which are negatively charged, and they're going to attract cations such as potassium, so that they'll want to start to enter into this potassium channel. Then as the potassium starts to travel up, it goes past the cytoplasmic opening, and we eventually end up in this central pore. The central pore is a little bit larger. It's 10 angstroms in diameter, and it contains these hydrophobic side chains that we can see in this diagram in yellow. And so this allows for hydrated potassium to go right through. And we can sort of think of this as similar but different with our aquaporin, right, where we had these hydrophobic residues lining it that allowed the water to kind of slide right past them. So you can imagine this potassium ion, if it's surrounded by water, can also sort of slide past these hydrophobic side chains. Excuse me. And then, oops, I'm so sorry. Okay, and then the next we have as the potassium first had entered the cytoplasmic opening, passed through the central pore, now it's going to arrive at this extracellular opening, which is the exit towards the outside of the cell at the top of this diagram. This is where the selectivity filter is located. So So one component of this is the diameter. It's very small, only 3 angstroms. And in addition to this, it has this conserved sequence of TVGYG. We'll see more about that in the next diagram. And this is where potassium is going to sort of change. In the central pore, potassium had to be hydrated to make it through, surrounded by water. But because of the size and this conserved amino acid sequence at the selectivity filter, the potassium essentially becomes dehydrated basically strips away the water as the potassium passes through, and that allows only potassium to eventually leave out the outside once it makes it to the other end. So let's look a little bit more in detail about that selectivity filter. So, excuse me. So you can see so here's this conserved sequence, this TVGYG up here. And so you can see that these side chains, or rather the backbones, the side chains are projecting towards the outside, right? And then we have the backbones that contain oxygens that are pointing towards the middle of that pore. Those are going to help interact with potassium itself, dehydrated. So it's going to, they're going to sort of form this cage that fits exactly around a dehydrated potassium, and that's going to help exclude water from coming through with it. And so ultimately, by the time the potassium passes through the other side, it is dehydrated. And then once it leaves, it will probably coordinate again with some water molecules. But to get through this selectivity filter, that potassium has to get dehydrated. And that's different from other regions, such as in the central pore that we see down here, where potassium is able to interact more strongly. It's surrounded by this sort of cage of water molecules that we see on the bottom there. All right. So that was our KCSA potassium channel. It's essentially always open, right? And it just it's specific for potassium, and it only lets potassium go one way because that's the direction down the electrochemical gradient for potassium, going out of the cell from the inside, where generally there's a high concentration of potassium. So next, let's consider these potassium voltage-gated channels. So these voltage-gated channels have a relatively similar pore to KCSA, which allows them to be very specific to potassium. And so, similar to KCSA, the whole voltage-gated potassium channel is a homotetramer, and it has one pore. It has 2 long helices, which are S5 and S6, which we can see in this diagram here, as well as over here. And then there's also short helices, that are the other numbers. And all those helices together help to combine to make this pore, which we can see here, where we have our little potassium ion right in the middle. And again, we have a similar selectivity filter that we saw with our KCSA channel. But in addition, we have more helices, S1 through S4, which we see over here, as well as the T1 domain, which is labeled over here. So we have our actual channel, which looks very similar to KCSA, and then in addition, we have this other component, which is the T1 domain and those S1 through S4 helices. So let's talk more about what they do. So to talk about what they do, we had to think about one of the things that's different about the voltage-gated potassium channels, which is that they are able to open and close in response to the membrane potential of the cell. So if we recall when we talked about defining membrane potential in the previous lecture, that's relative to the inside of the cell. So in a resting cell, it has a polarized membrane, and the membrane potential is about, negative 70 millivolts. And at this point, when the membrane potential is negative but polarized, or it's polarized, meaning that it's negative, this is when that voltage-gated channel is closed. Then during the course of what we call an action potential, that we'll discuss a little bit later, that frequently comes as a signal from your brain, an electrical signal, the membrane of a cell can go from polarized to depolarized, where the where the charge on the inside of the cell increases so far up past negative that it becomes positive, up to about 40 millivolts. And when the membrane is depolarized, that's when these voltage-gated potassium channels open. And then finally, the next stage after this, we have this hyperpolarization of the membrane, where the membrane channel decreases, again, goes down towards 0 and all the way into the negative, and it goes even further negative than it originally was. So its original state polarized about 70 millivolts. And then this hyperpolarized state after the depolarization, we have negative 80 millivolts, so it's even more negative. And at this point, the voltage-gated potassium channel is inactivated. So overall, the voltage-gated channel for potassium has 3 different states: closed, open, and inactivated. And these are related to the membrane potential of the cell membrane, so whether it's polarized, depolarized, or hyperpolarized. And the actual opening and closing is due to these different helices that we mentioned earlier. So first, let's look at what the potassium channel looks like when it's closed, when the cell is polarized. And again, this means that the inside of the cell is more negative than the outside. And we can see, based on this picture, the channel is very clearly closed. We can't have potassium ions coming in the bottom and exiting out the top because the channel is closed because of the movement of these helices around each other. When the membrane is depolarized, this is going to help open up the channel. And we can see here like the movement apart of these different helices from one another. This makes it open, and this literally makes a space that allows the potassium ions to pass through. And at this point, the inside of the cell, and like we said, this opening of the voltage-gated channel is happening during depolarization of the membrane, when the inside of the cell becomes very positive compared to the outside. And so like I said, we have this movement of the helices that helps to open up the membrane, and in particular, this S4 helix, which has a positive charge, is going to move up and away from the inside of the cell, which is now also positive because it's now more attracted to the outside of the cell, which is relatively negative. And we can see that also in the middle of this diagram, where we see this S4 and S5 helix that are all sort of being pulled up together, and that helps to open up and change the shape of this voltage-gated channel. And we finally see our open channel here on the right, where potassium ions are able to pass through. So that was our second step. So remember, we started with polarized, where we're closed. Then we were depolarized, channel open. Then the next step is the hyperpolarization of the membrane. At this point, the inside of the cell once again becomes negative, and it becomes even more negative than it was to begin with. And at this point is where the T1 domain comes into play, where there's this inactivation ball. And the inactivation ball actually blocks access to the potassium channel. And so if you can sort of picture this T1 domain, and here's our pore where the potassium ions are passing through. If you have something that's physically blocking this area, then potassium cannot pass through. So we have, not only is the channel it's not just closed, it's blocked. It's completely inactivated. So even if at this point the membrane potential changed, because you physically have this inactivation ball in the way, you cannot physically have ions passing through here. So let's talk about the context of this membrane polarization and depolarization. And so this is often due to nerve impulses, where we have nerve impulses that are propagated by the opening and closing of ion voltage-gated channels for both sodium and potassium. And this combination and the whole process of this cycle of the action potential helps to ensure that the signal from a nerve kind of travels in one direction, essentially downwards, instead of spreading all over the place, or it helps to give it essentially a directionality. And so let's talk about why this happens. So we have at the beginning state, right, we have our resting potential right around minus 60, maybe minus 70 millivolts. And this is our resting polarized cell. When we have an action potential, we see the membrane potential going up. It goes all the way past 0 into the positive until it reaches a height at around 30 or 40 millivolts. And that's the peak of our action potential. At this point, then it goes back down. We have so it goes from depolarized at the top, and then it goes back to polarized at the bottom. And then it goes back to depolarized at the top. And then it goes back to polarized at the bottom. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top. And then it goes back to depolarized at the top down to this hyperpolarized state. So it goes all the way down past where the resting potential was to this hyperpolarized state, around -70, -80 millivolts. And that's at that point where the channel is deactivated. And so if we think about this in terms of our voltage-gated potassium channels, at this resting state, we're closed. At our depolarized state, we're open. And at our hyperpolarized state, we are inactivated. And this is for our voltage-gated channels. Or voltage-gated potassium channels, excuse me. So let's think about how this action potential is actually mediated by the opening and closing of these sodium and potassium voltage-gated channels. So at the beginning of our action potential, we have a nerve impulse, and this is actually going to, so first we have to consider the normal states of sodium and potassium inside and outside the cell. So we already talked about how for potassium, it's normally found at very high concentrations inside the cell and very low concentrations outside the cell. So if you open a voltage-gated potassium channel, potassium will leave the cell. Sodium is the opposite. Sodium is present at higher concentrations outside the cell than inside. So if you open a voltage-gated sodium channel, sodium will flow into the cell. And so that's what we're seeing sort of here. So on the left, we had our measuring our membrane potential from our action potential. Now, if we look at this graph on the right, we can see how this is related to ion permeability through the opening and closing of channels. So as we go from our resting state to our depolarized state, we have an increase in sodium ion permeability, meaning that voltage-gated sodium channels are opening. This is allowing sodium ions to flow into the cell where they were originally at lower concentration. And this is going to help to bring the membrane potential more positive, right? You're bringing more positively charged ions into the cell. We're going to have this depolarized state. And so that's why we see this height of sodium permeability right where we saw this height of our action potential, because that's where the cell is at its most positive, where we're letting in the most sodium ions. But around the same time, during this during this depolarized state, this is when we also start to open our voltage-gated potassium channels, right? So we talked about how when the cell is depolarized, our voltage-gated potassium channels are open, and that's what we see on the right here with this purple part of our graph. When the voltage-gated potassium channels open, potassium flows out of the cell, right? And so this is going to make the membrane potential more negative again until you reach that hyperpolarized state at the end. And then once you've passed this hyperpolarized state, you have this period of sort of re-equilibration essentially, and once the cell membrane potential goes back to its resting potential, where it's just regular polarized, not hyperpolarized, then this whole cycle can begin again. But you have this resting period in between the action potentials, and this can sort of help make sure that the nerve impulses have a directionality and a timing to them. So excuse me. So let's talk more about that in detail. So we have our nerve impulses. It's being propagated by sodium and potassium voltage-gated channels. And so if you look at the bottom of this picture, we have our resting state. We're at our resting membrane potential, about -60 to -70 millivolts. We have a lot of sodium on the outside. We have a lot of potassium on the inside. And again, this is the resting state, where all of our channels are basically closed. So in yellow here, we have our potassium voltage-gated channel. In purple, we have our voltage-gated sodium channel. In the middle here, we have a different kind of transporter shown in blue. This is our sodium-potassium transporter. This type of transporter is also called a sodium-potassium ATPase. And one of the reasons for this is that it requires energy, right? See, we have our ATP. It's cutting ATP into ADP. That's why it's an ATPase, and it's using the energy of cutting this bond to phosphate to fuel this movement of sodium and potassium ions. So, and this is what helps. This particular sodium potassium transporter helps to maintain the homeostasis, this resting state, where you have a lot of sodium on the outside and a lot of potassium on the inside. So, like we mentioned before, if you just open one of these voltage-gated sodium or potassium channels, the ions are going to flow down their electrochemical gradient to where there is a lower concentration and charge of them. So we need this sodium-potassium transporter to restore that balance. So the sodium-potassium transporter is going to be pumping sodium ions outside of the cell so that there's a lot of sodium outside of the cell. And at the same time, it's going to be pumping potassium ions inside the cell so that you have that higher concentration of potassium ions inside the cell. And so we always need this sodium-potassium transporter to be working to help maintain that sort of resting state. So in this resting state, our sodium-potassium transporter is working to help maintain that homeostasis. And our sodium and potassium voltage-gated channels, like I said, are closed. So in response to a nerve impulse, now we have the beginning of this depolarization process where our voltage-gated sodium channels are going to open. This allows the sodium to flow into the cell, and this ultimately is going to help more of a positive charge be inside the cell, and this increases the membrane potential, so it's more positive. So it goes all the way from negative to 0 and all the way past 0 to about plus 30, plus 40 millivolts. And this is our action potential in our depolarization. Then in step 2, once we've had this depolarization, now the sodium channels are going to close. So sodium is no longer being allowed to enter the cell. And instead, our potassium channels, our voltage-gated potassium channels, are now going to open in response to this change in membrane potential, right? And this is going to let the potassium ions flow back out of the cell. So now you have a positive charge leaving the cell. And ultimately, this is going to help the membrane become hyperpolarized. So it goes all the way down from positive 30 or 40 to 0, and it keeps going all the way down to negative 70, negative 80. And then once the cell is hyperpolarized, now all of our sodium and potassium channels are inactivated using that inactivation ball that we talked about before. And during this sort of resting state where you're not having this movement of ions, the action potential is then going to physically move down the axon so that you have the movement of this nerve impulse in one single direction. And then after the state of hyperpolarization, the initial gradient of both sodium and potassium is then meanwhile being restored by the sodium-potassium transporter that's pumping away, pushing the sodium back out, and pulling the potassium back in. And then the channels, at that point, once the initial gradient is reestablished, we have the return to our normal resting polarized state, and the channels will return to their regular closed state as opposed to being inactivated. And so once again, as a reminder, the sodium-potassium transporter, it needs to use ATP because it's making the gradient. It's not just pushing the ions with their gradient, like I mentioned before. So these voltage-gated channels are allowing the ions to go down the gradient. Sodium-potassium transporter is making the gradient. It's pushing both of these ions against their gradient. And so that's why ATP is required. And this is a type of active transport, which we're going to cover more in the next lecture. So at this point, now you should be ready to answer all of the Chapter 10 passive-mediated transport practice questions.